Okay, everyone, we're going to move on to our next presenter, which is Nuria Rico. Nuria joined the Nekton team as their lab manager and is leading the zooplankton research, which I imagine means she spends a lot of time looking down a microscope. Nuria is a marine biologist and she's in love with zooplankton taxonomy. And she's dedicated all her scientific career to gathering as much information as possible about these fascinating creatures. So today she's going to talk to us about zooplankton biodiversity in the Seychelles. So I'm going to pass over to you now, Nuria.
Thank you, April. I'm going to share my screen. Yep. And then see that. Perfect. So, well, thank you for the presentation, April. And okay, so let's start with what zooplankton biodiversity in the Seychelles waters. So first of all, let's talk about what is zooplankton. Well, the definition could be animal drifters found in large bodies of water. So by large bodies of water, we're saying that we can find them in marine ecosystems and also in freshwater ecosystems. And by animal drifters, we're saying that they're able to swim, but they cannot swim against currents. So the zooplankton belong to the plankton community. And depending on how much time they spend in that community, we can divide them into two groups. Those that spend the entire life in it, which are called the holoplankton. And some examples are some examples are copepods and jellyfish. And other is those that only spend a little time of their life in the plankton community, and it's usually the first moments of life, which is the larval stages. And those are called the meroplankton. Some examples could be fish and crabs. So then we have this group of animals, and they're like really diverse, we have different sizes. So how do we scientists divide them into categories? So the most popular ways is by doing it by size, and some classifications are megaplankton, which is the higher rank, which is animals that are bigger than 20 centimeters. And then from there, we will go into smaller sizes and ranges. So macroplankton, mesoplankton, and microplankton. So to get an idea of the size of how small they go into, microplankton has a size range from 200 micrometers to 20 micrometers. So then another way would be by taxonomy group. So here we have four of the biggest phylums that we find in zooplankton, which is Arthropoda, Mollusca, Chordata, and Cnidaria. So then we have this organism that we have in front of us, and we say, ooh, it looks like it belongs to the phylum Arthropoda. And then from there, we go down into the phylum, so like the pyramid, we go down until we get to the point of saying hopefully, what kind of species we have in front of us. So why is it important, zooplankton? Well, there are the link between the primary producers, in our case, phytoplankton, and higher trophic levels in the aquatic food web. So by higher trophic levels, we could put an example as the big blue whale. Also, they participate in one of the biggest migrations that we have in the world, and it happens every day during sunset and sunrise, and that's the Deal vertical migration. Also, as we said in the previous slides, some higher trophic levels, they belong to the zooplankton during the first stages of life. So zooplankton is kind of like the nursery of higher trophic levels. They also support the biological pump through different ways of carbon export, such as their exoskeleton or the fecal matter, for example. So now let's go into the fun part, which is how do we collect the zooplankton in the Seychelles water? So as we see here, we have two images, one at day and one at night. We collected samples both times since we wanted to see how is the Deal vertical migration in the Seychelles water. So we drag our newsster net over the surface for 20 minutes, and then we collect it and we bring it up to the boat. Then afterwards, we take care of the code end, which is the end part of the net. So everything that has gone through the net ends up in the code end. So we need to be really careful since we have there everything that our goodness in a way. And then we put it in a bucket to protect it, and we will empty it into a sieve to concentrate all the sample that we have. And then after concentrating the sample, we're gonna split it. In our case, we split it into 4 samples, and for that we use a Folsom splitter, as we see here in the 2 images. So after splitting, we're gonna collect it into a falcon tube, as we see here in our picture, and we had 4 samples. So after, like, what I will take, one of those samples, it was for zooplankton analysis. So we had a lot of samples, and more or less they're all really crowded, as we see in the middle picture. So for analyze one sample like this, it will take a long time. So we have to split it again and divide it into other subsamples. And we end up with something like that. And you're like, there's really nothing there. Actually, there is. It's just a tiny portion. And that is the portion we're going to take to the stereo microscope. And there's when we are going to divide it into the groups. After the division, we're going to get something similar to this. So this is more or less everything that we can find in that little sample. In that little sample, I have to say that at least we have more than 100 copepods, which is these organisms that we have here, and they're the major dominant group in the marine ecosystem, the zooplankton marine ecosystem. And then at least more or less in all samples so far, we have had at least 16 different groups, and they reach really high numbers. So here we have 2 decapoda larvae with different types, and the decapoda are the shrimps, crabs, lobsters, and so on. In the middle, we have the gastropoda. We have here a butterfly, which is gorgeous, some fish larvae. And then my favorite one so far that I didn't know existed until I joined Nekton, which I'm really happy, is a halobate, and is the only true marine insect, which I thought it was really cool. So after having this analysis, let's go a little bit of what so far we have. So I have to say that these all results I'm going to show are preliminary. I haven't finished analyzing all the samples, and so yeah, it's kind of like I'm going to show you a little sketch of what so far we have. So as we see here in this graph, in this zooplankton composition, we have in copepoda, we have at least almost 11,000 organisms found so far in all the samples as in above. So this is something we expect since copepods are usually the dominant groups in the oceans. And then copepoda belong to the arthropoda. So like in a way, family resemblance family members in the group, we have at least 1,700, but they're not the highest number that we have, since the second group that we have more numbers is mollusca with at least 2,000. There goes fish and eggs, gelatinous organisms, and then in order we have other at least 15 different groups that their numbers were too low compared to the other ones, so I grouped them together for this presentation. So after doing a zooplankton composition, we wanted to check, okay, so is there a difference of abundance between sites? So our abundance is individuals per meter cube. And we did it for, first initially we decided to do it by regions. So we group the first, which will be the Amirantes region, which involves Alphonse, Poivre, Darros, and the Roches. And then the second region will be the Aldabra region, which involves Aldabra and Astove. So we checked it out and did a statistical analysis, and we did not find a statistical significant difference between the abundance between these 2 regions. But then we were like, okay, what about difference between all sites? And we did an analysis, and we did find a statistical significant difference, which was mostly driven by Astove that we have here. It was most of the time different with all the other sites, which will be something curious to look at in the future. Again, since these all preliminary results, these results I've just said, they're not really saying us a little bit what's going on, but we have to do further studies to look into it. And now we wanted to check, as I mentioned before, is what is going on in the dual vertical migration? Do we get something? Do we have a difference of abundance between day and night? So for that, we checked and we did not find a statistical significant difference so far in abundance from the night and day samples. So there was not a difference between what we had at night, which is when the migration comes up, And then during the day when the migration, the population of organisms is in the depths. But we still were curious and we were like, okay, we don't have a difference in abundance. What about in the composition? Like, is there a difference between different types of organisms? So we did this graph in which we have in the y-axis the number of organisms and in the x-axis the different groups. For day and for Copepoda, in between day and night, we did have a difference. There's more, we have more numbers in the day than at night. The same in mollusca, but not much in eggs, fish and eggs, gelatinous organisms, and the other organisms. So there's not much difference there. But then in Arthropoda, we have something really interesting. It is that during day, they're like a reduced number, but at night, it increases. So this is mostly driven by this, the Decapoda larvae zoea. And then the numbers are the ones that are making the increase at night. So after all this, I would like to say merci. Thank you for everyone for listening and everyone that participated in the expedition and collecting the Houston samples.
Thank you so much, Nuria. That was really interesting. We've got a few questions here. Have you considered the ingestion of microplastic by the zooplankton in your research? Did you see any of that?
No, I haven't considered the ingestion. We do some plastic particles in the samples, but we did not count for that in the beginning of the sampling, so we cannot really say much for now. We might have to think about it if we can implement it or so.
Yeah, something for the future.
Yeah.
And there is a question about the difference in zooplankton distribution between the monsoon seasons, but I understand you only were sampling during the northwest monsoon, so.
Yeah, we only went there last year, only for a period of 2 months. So it was like a period of time. So to be able to see, we have like a distribution or like the seasonal plankton, zooplankton is known to have seasons and like different. And for that, we will need to do like almost a sampling method almost every 2 months to get like something more robust.
Okay, great. And I have a question about the Decapoda. Were you able to identify them down to species level? I'm interested in the coconut crabs. It'd be really interesting to see if there's lots of coconut crab larvae around Aldabra, for example.
I would love to, but first I do it by runs. So this is the first run that I need to go through all the samples, and then I go into species level, which I call the second run, is to go into mod depth. So now I'm trying to like divide everyone into the box in a way, and then I'll go into detail in the box. But I would like to.
Excellent. A couple more quick questions. How frequent are the true marine insects on your samples?
They're kind of, I do see them, but not as often as I would like to.
And then just a last one. Why did you choose to combine the fish and eggs? Were you sure they were fish eggs?
I combined them in this study. Most of them are fish eggs. I do have some other eggs. You might see them of copepods and other organisms. But in this group, we're starting for the first, like I said, on the first round, I first compiled them, and then I put them together, fish and eggs, because the numbers were too low. Since I don't have all the data right now, so I had to do a little sketch for everyone to see, in a sense.
Okay, there are more questions, but unfortunately we've run out of time, so perhaps we can get those direct messages to you and Nuria to answer.
Thank you so much.
Thank you very much.